Introduction. Welcome to today's lecture on the topic of cellular stress and homeostasis. In this session, we will explore the impact of stressors on cellular physiology and delve into the intricate cellular responses that are activated to maintain cellular balance and survival. We will discuss various signaling pathways, gene expression changes, protein folding mechanisms, autophagy, cellular senescence, redox signaling, apoptosis, epigenetic modifications, mitochondrial dysfunction, immune response, and inflammation in the context of stress. Let's begin our journey into the fascinating world of cellular stress and homeostasis. Cellular stress triggers the production of stress proteins and the activation of signaling pathways. Chaperone proteins like HSPs aid in protein folding and prevent damage. Cells also activate detoxification pathways and remove damaged components through autophagy. Gene expression changes help cells adapt to stress conditions. Knowledge of these mechanisms is essential for developing therapies for diseases associated with cellular stress. The impact of stressors on cellular physiology is diverse and depends on various factors such as the type, duration, and intensity of the stressor. Stressors can disrupt essential cellular processes, such as protein synthesis, DNA replication, and cell cycle progression. They can also impair mitochondrial function, alter cellular metabolism, and induce DNA damage. Furthermore, stressors can activate specific signaling pathways, such as the mitogen-activated protein kinase, MAPK, pathway and the nuclear factor kappa-B, NF-kappa-B, pathway. These pathways play crucial roles in coordinating cellular responses to stress, including the activation of gene expression programs and the secretion of pro-inflammatory cytokines. The phosphoinositide 3 kinase, PIEK/ACK pathway plays a major role in cell survival and growth during stress. Activation of this pathway promotes cell survival by inhibiting apoptosis and enhancing protein synthesis. In conclusion, intracellular signaling pathways such as the MAP pathway, the NF-kappa-B pathway, and the PIEK/ACK pathway are essential for the cellular response to stress. Activation of these pathways leads to the regulation of gene expression and cellular processes, ultimately promoting cell survival and adaptation to stress. In summary, when cells experience stress, the way genes are expressed changes. Transcription factors, like HSF1, play a key role in regulating these changes. HSF1 binds to specific DNA sequences called response elements and activates the production of heat shock proteins, which help maintain protein function during stress. Protein folding is essential for protein function, but stress can disrupt this process. Cells rely on chaperones, like HSPs, to help refold or degrade misfolded proteins. Chaperones also regulate several cellular functions, such as signal transduction, cell cycle progression, and immune responses. Autophagy is an essential cellular process that degrades damaged or dysfunctional cellular components to maintain cellular homeostasis. During stress, autophagy is upregulated to remove misfolded proteins, damaged organelles, and intracellular pathogens. The process of autophagy involves the formation of double membrane structures called autophagosomes, which engulf cellular components targeted for degradation. These autophagosomes fuse with lysosomes, forming autolysosomes, where the cargo is broken down by lysosomal enzymes. Through autophagy, cells not only eliminate potentially harmful entities but also recycle cellular components to maintain energy production and survival during stressful conditions. Cellular senescence is a response to stress that halts cell growth. This process prevents damaged cells from multiplying, but too many senescent cells can lead to tissue problems and age-related diseases. It involves the activation of specific pathways that halt the cell cycle and promote the secretion of factors that can affect the tissue and immune response. Redox signaling plays a crucial role in the cellular response to stress. Under normal conditions, cells maintain a balance between oxidants and antioxidants, known as redox homeostasis. However, during stress, this equilibrium can be disrupted, leading to an increase in reactive oxygen species, ROS, levels. ROS, such as superoxide anions and hydrogen peroxide, can act as signaling molecules that regulate various cellular processes including cell survival, proliferation, and apoptosis. ROS levels are tightly controlled by antioxidant enzymes, such as superoxide dismutases and catalyses, which scavenge free radicals and prevent oxidative damage. Apoptosis is a regulated process that removes damaged cells during stress. Proteins like Bax and Bach promote cell death by releasing cytochrome C, activating caspases to dismantle the cell. Proteins like BCL2 and BCL-XL inhibit apoptosis by preventing cytochrome C release. Balance determines cell fate. Epigenetic modifications are changes in gene expression that are not due to alterations in DNA sequence. DNA methylation, a prominent epigenetic modification, involves the addition of a methyl group to cytosine residues, repressing gene expression. Stress can alter DNA methylation patterns, affecting gene expression and cellular behavior, and contributing to diseases like cancer and neurodegenerative disorders. Mitochondria, the vital cellular organelles responsible for energy production and maintaining cellular homeostasis, can be negatively affected by stress. This impairment results in the generation of harmful molecules, disruption of energy production, and the release of factors promoting cell death. Various stressors such as nutrient deprivation, oxidative stress, or alterations in genes can induce mitochondrial dysfunction. These impairments often encompass structural and functional changes in mitochondria, including energy production, division, and fusion processes. In response to mitochondrial stress, cells employ mechanisms to restore normal mitochondrial function, such as generating new mitochondria and eliminating damaged ones. Stress can significantly impact the immune system and initiate an inflammatory response. The immune system detects cellular stress signals and triggers the activation of immune cells, such as macrophages and lymphocytes, to combat potentially harmful agents. Upon stress induction, cells release pro-inflammatory cytokines, such as interleukin-1 beta, IL-1 beta, and tumor necrosis factor alpha, TNF-alpha. These cytokines attract immune cells to the site of stress and promote inflammation. Inflammatory response acts as a defense mechanism against pathogens, but chronic inflammation can lead to tissue damage and contribute to the development of chronic diseases, including cardiovascular diseases, autoimmune disorders, and neurodegenerative diseases. To effectively combat stress-related diseases, it is crucial to understand the intricate mechanisms by which cells respond and adapt to stress.